The overall goal of this single-molecule force spectroscopy technique is to determine the adhesion force and nature of interactions between amino acids or peptides with different surfaces.
This method can help answer key questions in the area of surface chemistry and be used for the development of new composites and coatings. The main advantage of this technique is that it provides information on interaction between the desired molecule and surfaces at the single molecule level.
To begin, purchase silicon nitride AFM cantilevers equipped with silicon tips with a nominal cantilever radius of 2 nanometers. Mount the tip on the holder. Clean each of the AFM cantilevers by dipping them into ethanol for 20 minutes and then dry them at room temperature. Next, place the tips into a plasma chamber and treat the cantilevers by exposing them to oxygen plasma for 5 minutes. Prepare a solution containing 15 parts methyl triethoxysilane and 1 part 3-amino propyl triethoxysilane and suspend the cantilevers 3 centimeters above the solution while under an inert atmosphere.
It is important that the tips and the holder do not touch the silane mixture. In addition, the silane is highly moisture sensitive and therefore these steps needs to be carried out in an inert atmosphere.
Once sealed, connect the desiccator to a vacuum pump and apply a vacuum for 2 hours to form a monolayer of the 2 types of mixed silane compounds on the surface of the cantilevers.